And so again, good morning. So for today, we will be talking about flow cytometry and laboratory automation. And for today's topic, we will be subdividing it into the following. So under the flow cytometry, we will be discussing about instrumentation, data acquisition and analysis, sample preparation and clinical applications, and we will also be discussing on immunoassay automation. Okay, so let's talk about first cell flow cytometry. So the term cytometry is actually divided into two meaning, no? So the cyto here means cell and metry refers to the measurement. So the literal meaning of the term flow cytometry is cell measurement. So this particular cell measurement is considered to be as an automated process. So there is actually an instrument or a machine that can actually do this. And this particular machine can actually analyze cells. Okay, so you can actually analyze individual cells, or you can actually analyze beads. Okay, so sometimes you are using beads instead of cells, and this particular beads can be labeled, let's say, for example, with a fluorescent. Okay, so these particular cells are being suspended in a fluid suspension. So we call this as fluidics. Okay, for their light scattering characteristics. Okay, so there are actually two types of light scatter in flow cytometry. So we can have the forward scatter, and the other one is the side scatter. So later on, we will be discussing what's the difference between the forward scatter and the light scatter. Okay, so flow cytometry utilizes fluorochromes. Okay, okay, so when you say fluorochromes, these are fluorescent-based compounds. So these particular fluorochromes can identify cells or particles by means of size, okay, shape, and even antigenic properties. Okay, so antigenic properties. So when you say antigenic properties, it means that we can determine the cell surface markers. So if you say antigenic properties, we can determine cell surface markers. What are examples of cell surface markers? For example, if we're talking about T helper cell, the cell surface marker is CD4. Or if you are talking about cytotoxic T cell, then the cell surface marker is CD8. So this can also be identified in flow cytometry. So the main advantage of flow cytometry is that it allows rapid and accurate detection of cells even if they are found in small numbers. Okay, so that's the reason why this particular type of instrumentation is commonly being utilized in, let's say, for example, in hematology. So majority of hematology analyzers would also have the principle of cell flow cytometry. So cell flow cytometry is actually being used for the diagnosis or identifying HIV infection, particularly particularly in determining the CD4 CD8 ratio, because normally the CD4 CD8 ratio is 2:1. However, if you are now in clinical age, there is what you call the reverse CD4 CD8 ratio. Usually, the reverse CD4 CD8 ratio happens when your CD4 level falls below 200 times 10 to the 9 per liter, because the normal value of CD4 is between 800 to 1000 times 10 to the 9 per liter. So CD4 CD8 ratio is actually reversed if you are in the clinical age already, because it means that your CD4 level becomes significantly depleted. Cell flow cytometry can also determine immunophenotyping of cells. So like what I've told you, we can identify different cell surface markers. Not only that, we can even identify the glycoproteins found in platelets. So for example, you want to determine the glycoprotein 1B9 complex or glycoprotein 2B3A complex. This would be made possible by doing the flow cytometry. Okay, so cell flow cytometry can also be determined or can also be used for the diagnosis of leukemia, lymphoma, and functional assays for various conditions. So for example, if you are deficient with glycoprotein 1B9 complex, so you have a condition known as the Bernard-Soulier syndrome, or if you are deficient with glycoprotein 2B3A complex, then the condition is known as the Glanzmann thrombasthenia. So I hope you are familiar with this in hematology too. So these are the things that you're supposed to be discussing in hematology too. So um, various assays can be identified. Okay, various clinical conditions can be identified. And perhaps one of the most common um, purpose of cell flow cytometry would be for the WBC differential count. So cell flow cytometry can also be utilized in WBC differential count. Okay. So let's talk about the part of the instrumentation in flow cytometry. Okay. So the first thing that we should be discussing is about fluidics. So a while ago, we have already defined the fluidics. These are actually the cells being suspended in a liquid suspension. Okay. So here, cells are actually being suspended in a liquid suspension. So this is what you call the fluidics. And then laser light source. So you can see here that this is now the laser light source. Okay. Laser light source. Okay. Will identify two main parameters. Okay. The first parameter is the intrinsic parameters. And the intrinsic parameters can identify can identify two light characteristics. Okay, so if the light goes this way, this is what you call the FSC. Okay, the forward angle light scatter. And sometimes light will go at the other direction or it will go at the side. So this is what you call the side scatter or the SS. So there are two types, okay? And then we also have the extrinsic parameters, okay? So the extrinsic parameters are actually being utilized to determine, for example, cell surface markers, okay? Cell surface markers. And in order for these to be determined, we're using a fluorochrome fluorescent labeled antibodies. Okay, that will be made part of the fluidics. So, for example, you want to determine CD4 in the cell. Therefore, you should be able to use a fluorescent labeled anti-CD4. And then this particular fluorescent labeled anti-CD4 will actually attach to your CD4 cells, and then it will be determined here in the fluorochrome in the fluorescence detectors. So if you're using, as part of the fluidics, fluorescent labeled anti-CD4, and if you have CD4 in your cells, the fluorescent labeled anti-CD4 will be attached at the surface, and that will be determined as well in the flow cytometry or in the flow cytometer. So that way, we can determine also cell surface markers here in flow cytometry. And of course, there are several types of detectors. Okay, so what are the several types of photo detectors? We have the forward angle light scatter detectors. We have the side scatter detectors. And then we have the fluorescence detectors. Okay, we have the fluorescence detectors. Okay, so take note that we have identified 3 detectors. And even before this light will go to the detectors, there are also filters. There are also filters so that it will actually concentrate the light on the detectors. Okay, so these are the components of a flow cytometry. Okay, so all of these detectors will be generated in the computer, and then the computer printout will tell you the population of the cell. Okay, so this is an example of a single parameter histogram. By the way, histogram is actually the graph that will study the number or the cell population. Okay? So if the peak is high, so this is the peak. If the peak is high, this determines the cell count. So more or less, the cell count is here. Okay, so the peak depends on the fluorescent intensity. This would be applicable if you are trying to determine cell surface markers. So this is what you call a single parameter histogram. Now we have here a dual parameter histogram. Okay, so this is an example of a dual parameter histogram. So in the dual parameter histogram, I'll just review. In a dual parameter histogram, this can determine the side scatter. Okay, so we have here the side scatter, and then we also have here the forward scatter. Okay, so what does a forward scatter determine? Okay, so forward scatter will actually determine, the forward scatter will actually determine light scatter that has gone up 180 degrees centigrade. Okay, so that kapag 180 degrees centigrade, if yun nag-forward lang siya, that is actually a side scatter. So the other one measures light at 90 degrees centigrade. So balikan natin ha. So kapag 180 degrees centigrade, nag-forward lang siya, that is the forward scatter. Okay, so the other one is the light scatter, side scatter. Ibig sabihin, it measures the angle at 90 degrees. So it measures the angle at 90 degrees centigrade. Okay, so that's the basic difference between the forward scatter and light scatter. Okay, so take a look at this particular graph. No, this is the SSC parameter, and then this is the FSC parameter. No, take note that FSC measures the diameter of the cells. Okay? Whereas the SSC measures the internal complexity. So what do you mean by diameter of the cell? FSC, therefore, will determine the size. SSC, since it determines internal complexity, it will determine the granularity. Okay. So, ibig sabihin, guys, the larger the size of the cell, so for example, if this is the histogram, kapag larger pala yung size ng cell, it will go at this particular portion. Kasi ito yung FSC, ito yung SSC. So, nandito siya sa part na to. The smaller the size, nandito sa part na to. Okay? So, if this is the FSC, para hindi kayo malito, kapag large yung cell, nandito sa side na to. Kapag small yung cell, nandito sa side na to. Okay. Remember that there are two types of WBCs, right? WBCs could be granulocytes or agranulocytes. Granulocytes, ano ba yung mga granulocytes? These are your neutrophils, basophils, and eosinophils. They have granules. Ibig sabihin, since they have granules, then they can be detected also by the SSC, because SSC refers to internal complexity. And when we say internal complexity, it determines whether the cells have granules or not. So kung wari, ito yung SSC, kapag mababa yung portion, ibig sabihin, walang granularity. Pero kapag medyo nasa upper portion, ibig sabihin with granules. Okay. And then, tandaan ninyo in histogram, more dots represent increase in cell population. So kapag mataas yung, kapag marami kayo nakikita na dots, it means that there are more cell population. Now, let's talk about the dual parameter dot plot. So this is what you call the dual parameter dot plot. Why did we say dual parameter? Because here we have the SSC and here we have the FSC. Now take a look at RBCs. RBCs do not have granules. So that's the reason why at the SSC, it is more at the lower portion. And then the number of and then in terms of size, RBCs are smaller than WBCs. So that's the reason why nasa bandang dito lang siya. Nandito lang siya because it is not as, in terms of size, it is smaller. So it doesn't have granules and it's small. Okay, now take a look at neutrophils. Neutrophils, this is the neutrophil. Okay, neutrophil has granules. So that's the reason why at the SSC, it is at the upper portion. Diba? This is neutrophil, it is at the upper portion because they have granules. But take a look at monocytes and lymphocytes. Both monocytes and lymphocytes are considered to be as agranulocytes. They do not have granules. So at the SSC part, they are even actually at the lower portion. Now, when it comes to size, the biggest WBCs are supposed to be the monocytes. They're even bigger as compared to the neutrophils. So in terms of size, so neutrophil is here, and then monocyte is here. So this is now the dual parameter dot. So you can determine the size, and you can determine the internal complexity. Okay, so then take a look at the intensity. Lymphocytes and monocytes. Lymphocytes are darker as compared to monocytes, meaning to say there are more lymphocytes as compared to monocytes. And take a look at neutrophils. It is much darker as compared to the monocytes. So meaning to say there are more cell population of neutrophils than the monocytes. Okay, so this is how we analyze. Okay, this is how we analyze the dot plot in flow cytometry. Okay, and then if you are using a computer, you can use your cursor and study this particular cell. You call it gating. Okay, you call it gating. So the gate is drawn around the cells of interest. So let's say you're interested to study the neutrophil. So you draw the line here to study the neutrophil more. Okay, to a more detailed study of the neutrophil. Okay, now after you have gated, you can actually study the cell at a much detailed parameter. Okay. Let's say, for example, you have already gated the cell, and let's say, for example, this time you want to determine the cell surface marker, and you want to study three types. You want to study the population of CD3 and CD4. So what will you do if you want to study the population of CD3 and CD4? You apply fluorescent label. So for example, you want to study the CD3, then you need anti-CD3 FL1, fluorescent labeled one. And let's say, for example, you want to study CD4, then you use anti-CD4 FL2 or anti-CD4 fluorescent label 2. And then we can do the quadrant analysis of the dual parameter dots. So this is how it looks like, no? What does it mean? This particular cell is positive for FL2, okay, but negative for FL3. So ibig sabihin, yung cell na to is CD4, positive, CD3, negative. Makita niyo yung color na to, it's a combination of blue plus red. So anong ibig sabihin na to? So this cell is positive for CD4, and it is also positive for CD3. Now look at that quadrant number 3. Quadrant number 3 doesn't have any fluorescent label. So this is CD4 negative, then this is CD3 negative. Look at quadrant number 4. It is only positive for CD3, but it is negative for CD4. So this is now how we study quadrant analysis of a dual parameter dot plot. So, however, when you go to histogram, it doesn't look this way. This is the actual picture of the histogram. So, what can you say about this one? So, the CD4 population will be analyzed, will be counted by the computer. The dots will be computed by the computer, and it will be proportioned according to the number of cells in the entire picture. So this time, according to computer, it is 2.24%. See? Now, this one is CD4 positive plus other lymphocytes. And this accounts for 24.4%. Okay? These cells are the unidentified cells because they are neither CD4 or nor CD3. So this one is unidentified cells. Now this is the population of CD3. So the population of CD3 is 37.45%. Okay, so this is how we analyze. Okay, this is how we analyze the cells in Quadrant Analysis Dual Parameter DATS. Okay, so question, when do we use fluorescent label? Okay, we are using fluorescent label if we want to study cell surface markers, such as, for example, in this particular picture, our examples are CD4 and so therefore in summary, we can determine side scatter if we want to determine internal complexity. Forward scatter if you want to determine the size or diameter of the cells. With that, you can also do gating if you want to determine a specific cell population. And if you want to determine cell surface markers, then you need to use a fluorescent labeled antibodies. Okay, so for example, you want to determine CD4, then you have to include as part of the fluidics the anti-CD4 fluorescent label 2. Okay, so that's how we analyze the cells in flow cytometry. So what kind of samples do we study? We can actually study whole blood, particularly if you're drawing the blood intended for CBC. So it should be collected into a DTA tube. The sample is to be processed within 30 minutes or 30 hours. Okay, it should be stored at room temperature and should be well mixed. Take note not to utilize a hemolyzed sample. Okay, now if you really want to determine the cell surface markers of different WBCs, okay, or of different blast cells, okay, so we can still use peripheral blood. For blast cells, especially for the diagnosis of leukemia, okay, we can actually use bone marrow. So if you do not need RBCs, because you really want to study the WBCs, then some technique would even require erythrocytes removal. Okay, for tissue specimen, it should be collected and transported into tissue culture medium. So it may be stored at room temperature for imminent analysis or for immediate analysis. However, if analysis is to be delayed, then it is imperative that you store it at 4 degrees centigrade. So when you say 4 degrees centigrade, we are referring to the refrigerator temperature. So tissue specimens should be disaggregated via mechanical dissociation or enzymatic digestion. And we are actually using monoclonal antibodies before processing. Okay, so clinical applications of flow cytometry. We can use flow cytometry to identify a particular marker, especially intended for the diagnosis and monitoring of leukemia and lymphoma. So if you want to enumerate peripheral blood CD4 T cells to classify stages of HIV disease and guide treatment. So remember, you are considered to be as in the clinical age if your CD4 level is below 200 times 10 to the 9 per liter. CD34 positive cells can also be identified. Why? CD34 positive cell is an active cell. It is considered to be as cells that are pluripotential. Since it is pluripotential, then the enumeration of CD34 positive cell is important for stem cell transplantation. The flow cytometry will also determine the DNA content or ploidy status of tumor cells and help diagnose inherited diseases. So as you can see, no, there are many clinical applications of flow cytometry. Okay, so in other topic, we have immunoassay automation. immunoassay automation reduces errors. Then it is more accurate and precise and requires fewer staff. So it saves on control, duplicates, dilutions, and repeats, and has a potential for better sample identification using barcoding. So barcoding nowadays, particularly for immunoassay, is now the trend. So there are actually two types of analyzer that we can use in immunology. So the first one is called batch analyzer. So when you say batch analyzer, this one is very efficient because it can examine multiple samples and it provides access to samples for formation of reaction mixture. Um, However, even if we can determine multiple samples, it will only permit one type of analysis at a given time. Okay, so random access analyzers can measure numerous analytes from multiple samples. Oh, this one is much better. Okay, it can perform different tests on any one sample. Okay, so I hope you can actually determine the difference. Okay, so when you say batch analyzer, several samples, one type of test. But when you say random access analyzers, it can be one sample, different types of tests, or it could be different types of tests on a different types of samples. So that's the difference between the batch analyzers and random access analyzers. So there are several things that are being done by the automation during the analytical stage of testing. Imagine the machine will do the following for you: introducing the sample, aspiration of the sample. When you add the reagents, hindi na kayo magpapipette, mixing reagent and sample, incubating, detecting, calculating, and reporting. All you have to do is to copy the report. Prayerfully, you'll not commit any clerical errors when doing so. However, because of the laboratory information system, okay, the possibility of having clerical errors are also now being somehow eliminated because of the laboratory information system. And of course, whenever we are doing immunoassay automation, we want to have accuracy. Accuracy is the test ability to measure what it claims to measure. Meaning to say, the closeness to the true value, that is accuracy. The ability to consistently produce the same result on repeated testing of the same sample, that's the definition of precision. Okay, so if the bullseye is the true value, this is how accurate and precise sample would look like. This is how an imprecise and inaccurate sample would look like. This is how a precise sample but inaccurate result would look like. So take note, a precise result does not necessarily mean an accurate result. However, accurate results are always precise results. Okay. So this time, when it comes to diagnostic validity, we have already discussed what do we mean by analytical sensitivity. This was our lesson from last week. We have already computed how to determine this one. So when you say sensitivity, it is the lowest measurable amount of an analyte. So this was our lesson from last week. I hope you can still remember it. When you say specificity, the assay's ability to generate a negative result when the analyte is not present. When you say reportable range, the range of values that will generate positive result for the specimen assayed by the test procedure. So sometimes if the assay is not sensitive, so the reportable range would be very high. Okay, so reportable range, hanggang saan lang ba pwedeng ma-measure? So that's what we meant by reportable range. However, it is different from reference interval because when we say reference interval, it is the range of values found in healthy individuals. So it's the normal values who do not have the condition that is detected by the assay. So Reference interval used to define the expected value of a negative test, meaning to say you do not have the health condition that is being detected. So, for example, that particular assay will test hepatitis B surface antigen. And if you do not have hepatitis B, then you will fall within the reference interval. Okay, so this ends the first part of our lecture for today.